Hi everyone, it's 10 o'clock here in Montana, so we're going to go ahead and get started. I'm Cheryl Rogers, the Director of Marketing here at Golden Helix, and our webcast today is presented by Bryce Christensen, who's our Director of Services, and he will present genomic prediction. Bryce, the floor is yours.
Okay, Cheryl, thank you very much for the introduction, and I'd like to thank our audience for joining us here today for this very interesting topic that we'll be talking about with genomic prediction and how it can be done within the Golden Helix SNP and Variation Suite software. If you have any questions at all during the presentation, feel free to enter them in the GoToWebinar question panel. We'll keep an eye on those and try to answer as many of them as we can at the end of the presentation today. Today we're going to learn a little bit about the Golden Helix SNP and Variation Suite. I imagine that many of you joining us today are already familiar with SVS. I'll just point out that SVS is a platform that allows you to work with many different types of genetic data, particularly genotype data, either from a GWAS type of setting or also in a genomic prediction environment. You'll find that SVS has very useful and powerful features for data management, visualizations, robust and fully supported statistical methods. And as we go along today, we'll see a flavor of some of that and learn how it can be used for genomic prediction as well. So the outline for today's presentation will go something like this. We'll move between these topics a little bit, but we'll start by discussing some of the basics of genomic prediction, why it would be used, and what can really come of it. We'll mostly focus on agricultural applications today. Of course, there are applications in other areas as well, but we'll start with a case study based on cattle production, and then we'll move into a plant-based case study where I'll use some Arabidopsis data that I have access to. Finally, we'll move on and look at an interactive demonstration with SVS and learn a little bit more about the SVS environment and see just how you would go about doing this kind of work within the software. So to begin with, genomic prediction in recent years has become more and more important as a focus for plant and animal research and for agrigenomics in general. A lot of this can be explained by growth in the worldwide population. This, of course, requires us to improve our food production techniques. Yields need to be increased. We need to find methods that are more environmentally sound and more sustainable to support future population growth. Population has grown about 150% since the 1960s. It's expected to increase by another 2.5 billion or so in the next 35 years. And this is a major burden and a major concern that we all need to be aware of. And one of the methods that is being used to drive these improvements is genomic prediction. So why use genomic prediction? On this slide, I'm going to mention 3 different scenarios where it might be useful or important. First of all is this concept of calculating breeding values for subjects in a population. And what do we mean by breeding values? Well, historically, traditionally in breeding programs, you might rely on pedigree data, observational data about animals or about plants to determine which ones are optimum to breed for particular traits. You know, looking at these 2 cows on the bottom, if you're interested in producing larger cows, historically you might say, well, obviously this one is bigger, that's the one that I will breed. However, if we look at the genetics, we might be able to determine, you know what, that cow's so big because it got a little bit lucky. There's a cow that's just a little bit smaller that really has a better overall combination of genes that's more likely to produce similarly sized cattle in future generations. So that's one way we might approach genomic prediction. Another would be predicting those same types of breeding values for subjects with unknown phenotypes. So with the cattle example, yeah, we can look at them and say that one's bigger than that one. But what if you're looking at something that is more difficult to measure? Maybe you have been involved in a breeding program where you've developed new hybrid varieties of rice or wheat, and you're interested in determining which of those varieties will have the best protein yields going forward. That might require costly, lengthy field trials to go out and grow these all to maturity and do some testing on them. But with genomic prediction, if we have a good reference population where we can say, historically these various genes have produced these types of yields, we can get some really good predictions and avoid that process. Also, going back to the cattle, sometimes it might not be possible to measure the phenotype. Maybe you're looking at a meat quality trait that can't be measured well while the animal is still living, or perhaps even you're looking at milk production, but you need to select a bull, and of course you can't determine milk yields from a bull, but you might be able to build a genetic model based on the female cows to determine which genes are associated with better production and thus select the appropriate bull. And then a third approach for genomic prediction is simply to identify which genetic markers have the best predictive power for a trait. In that sense, it's very similar to GWAS. We may have hundreds or thousands or even millions of genetic markers and want to determine which ones should be used for decision making, and genomic prediction can help us determine which SNPs in which combinations are most appropriate for developing predictive tests or diagnostic assays and so forth. So the first method that we're going to look at today is GBLUP, or Genomic Best Linear Unbiased Predictors. Just a few things to be aware of regarding GBLUP is that, to begin with, it assumes that all loci contribute to the phenotype. So again, whether you have hundreds or thousands or millions of genetic loci available, it will try to include all of them into a model. Now that model is based on a mixed linear model framework. It incorporates a genomic relationship matrix, or GRM, which helps to account for relatedness among samples and make sure that no group or cluster of samples is too influential in the results of the prediction. In terms of output, it gives what we call an allele substitution effect for each SNP. Essentially, that is the coefficient of each SNP as it is built into that linear model, and tells you what is the effect size associated with the SNP, of course. And also it gives the breeding value that we talked about on the last slide. So you can see one animal at a time, which are likely to pass on the best traits to their offspring. It also gives some other statistics of interest, including trait heritability estimates and a breakdown of variance components of the trait, the genetic contribution and error contributions. So the other method that we'll look at today is the Bayes-C and Bayes-C-pi family of methods. Now, these are brand new in SVS, included in version 8.3, which was only made available a few days ago. Now, the Bayesian methods are distinctly different from GBLUP in a few different ways. One of the major things to be aware of is that it uses a sampling procedure and a Markov chain Monte Carlo procedure to identify an ideal model based on usually a fairly small set of SNPs, as opposed to GBLUP that is including all SNPs in the model. Bayes-C and Bayes-C-pi will have something that is greatly reduced. Now, anyone who's familiar with Bayesian MCMC methods will know that that also comes at a computational price. It is going to run just a little slower than GBLUP, but as a result, you may get more concise estimates in some cases. Now that pi parameter, when you hear Bayes-C and Bayes-C-pi, what pi is the probability that any given SNP will have no effect or negligible effect on the phenotype, and that explains the difference between the two methods. Bayes-C has a fixed value of pi, while Bayes-C-pi allows that pi value to fluctuate, and it is ultimately estimated in the final model. Now, both of these, similar to GBLUP, will return allele substitution effects, estimated breeding values, and some other parameters. And an important thing to be aware of is that the SVS implementation of Bayes C and Bayes C-pi incorporates a genomic relationship matrix similar to GBLUP. There are some implementations of these methods that do not, so it's just important to note that. So for our first case study, we're going to look at some cattle data. Now, in this simulation, we're using real genotypes. It's based on 472 animals from the Bovine HapMap project, Illumina 50K genotypes, and using a fairly simple trait simulation. There are 5 SNPs simulated to have independent additive effects. You can see down here on the bottom this GWAS result, where we can recognize exactly where those 5 trait loci are. Some have very strong associations, some more moderate. But what we'll do with this is walk through and show some examples of what happens analyzing this data in a few different ways. We'll look at running GBLUP in SVS, Bayes C-pi in SVS, and a few comparisons of Bayes C-pi implementations available in the R package. Now, in these first few slides, what we'll have is an example using entirely within sample predictions. We're using all of the 472 animals as the training set, and then calculating breeding values and allele effects for the entire set of data. So to begin with, let's just look at those allele substitution effects and how they compare. So in this top panel, we see the negative log 10 p-values from a mixed model GWAS analysis. And again, we see 5 distinct regions that are associated. In the next figure, we see the allele substitution effects from GBLUP. And these two figures are really quite similar, maybe a little more noise in GBLUP than in the mixed model p-values up here. But generally speaking, you can see that with GBLUP, every SNP has some kind of effect associated with it. Some are very small effects, some are very large effects, but for the most part, they are non-zero. That is in contrast to what we see with Bayes C-pi. So again, this is several thousand SNPs across the genome, and we see 1, 2, 3, 4, 5 individual SNPs that are predicted to have very large effects on the trait. Maybe another one down here with a marginal effect, but the majority of SNPs have been reduced to a negligible near zero effect on the trait. And so this is the result of that simulation process with the entire MCMC process. It's attempting to identify the ideal set of SNPs to give the most efficient prediction model. Now, what happens with the actual predicted outcomes? On this first slide here, we get an idea of what happened with GBLUP. The figure is showing the actual phenotype, and we'll suppose that this is a meat quality phenotype in the cattle. One thing you'll notice when you compare the actual phenotypes with the breeding values is, as expected, the higher values of the phenotype have the highest breeding values. There is a strong linear correlation here, but you'll notice that it's not perfect. For example, the animal with the second highest breeding value, if you really look at it, is probably about the 5th highest measurement of the trait. And this is one of those examples where there's, of course, noise in the real phenotypes, in the observations, but if you look at the genetics, it means this animal has the best, or the second best actually, overall combination of genetics for the trait based on what's observed within the population. Now, in this case, GBLUP actually does a fairly good job of estimating the variance compared to what was simulated, really quite close overall. Now, what about Bayes C-pi? Bayes C-pi has some differences, of course, in how it works. And what we're seeing here on the left is a representation of that MCMC process, where initially it starts out incorporating several thousand markers in the estimation with non-negligible effects. But as it continues through the process, eventually it starts to converge on a very small number of SNPs. In this case, we saw there were 6 or 7 that had non-zero effects for the most part. And comparing here, you see the curves are not identical, but again, there's a randomness incorporated into the process. We don't expect them to be exactly identical, but both SVS and R, as we compared them, converge on a similar set of markers. And like we saw with GBLUP, there is a fairly strong linear correlation between the observed phenotype. Here it's been standardized, but it's still the observed phenotype compared with the breeding value. Now, just an interesting detail that came out as we were comparing these, and it's something to be aware of whenever you're using a simulation process. As you compare the allele substitution effects from 2 implementations of essentially the same method, you'll notice majority of the SNPs are near zero, and there are thousands and thousands of SNPs in this little splotch of blue here at the bottom left. There are 4 SNPs that have near identical substitution effects, then there are 2 that appear to be a reflection of each other. And this is a case where in the input data there were 2 SNPs at the same locus on chromosome 20. They're very strongly correlated with each other. The linkage disequilibrium R² was about 96%. And the 2 methods converged on different options. It's not a big deal. Essentially, we're getting near identical predictions between the 2, except a handful of cases that are off the diagonal. And those are all individuals where the genotypes were not identical at the 2 SNPs. So 96% LD is very strong correlation, but there are a few differences. And that just enters a little bit of noise into the prediction, but ultimately very, very similar, and we're happy to see that. Now, when you compare the 2 SVS methods, Bayes C-pi versus GBLUP, as we saw with the substitution effect plots earlier, GBLUP assigns some non-zero effect to almost all of the SNPs. We see this range up and down here, even though Bayes C-pi is putting most of them near zero. And it's reassuring to see that the SNPs selected by Bayes C-pi are generally among the larger effect SNPs in GBLUP as well. And the breeding values, which is what we see here, this is a direct comparison of the breeding value estimates, again have a strong linear correlation. You're not going to pick a very different set of animals for breeding using one method or using the other. So that was an example of some within sample calculations, just looking at breeding values and substitution effects. Now I'd like to look a little more at out of sample prediction performance. And for these next few examples, we're going to look at a 5-fold cross-validation procedure. For those who may not be familiar with how this works, we take the complete data set and split it into 5 random subsets and go through 5 rounds of prediction, where in each round one of those subsets is the test data while the other 4 are used to train the model. This sort of a process is very useful when you just want to estimate the predictive potential within a data set. You can do this several times, go through multiple rounds of cross-validation and produce the data necessary to calculate ROC curves, sensitivity, specificity, that kind of thing. For today, we're just going to run a single round of cross-validation and just take a look at the types of predictions that we get and look at how you might use them. So to begin with GBLUP, what we're seeing here on the screen is the allele substitution effects calculated within each of the 5 subsets. Of course, there's a lot of overlap between all of these, but it is good to see that they generally agree on where the major signals are. Now, moving on, here we get the predicted versus actual phenotypes. So here the predicted phenotypes are going to be calculated without having that sample included in the model building phase. So these are out of sample prediction. And as we look at this, the correlation isn't perfect, but it's far from random. In this case, the R squared, or the proportion of variance in the actual phenotype explained by the predicted phenotype, is about 26%. And in terms of a linear regression p-value comparing the two variables, it's very highly significant. Now, one thing you'll notice, and this is pretty standard from what I've seen in prediction in general, is that the more extreme values aren't predicting as well as what's in the middle. It's hard to get really good predictions on the extreme things, especially with smaller sample sizes where the higher values may not be well represented in the training data. Now, what about Bayes C-pi? Here we're looking at the substitution effects from 5 runs, and you'll immediately notice here that one of them converged very well on 5 SNPs. The others did not, and I should point out that I was doing some testing when this was being put together, so they all had slightly different parameters in how they were run. I wouldn't necessarily expect them to converge identically, but it's interesting to see what happens in this case, and let's see that on the next slide. Overall, the prediction performance here was slightly better than GBLUP. We see R squared of 37% versus the 26% that we saw with GBLUP. But if you stratify this and look at each of the 5 subsets, and especially that one that had the good convergence, that one with the strong convergence here is in red, you'll notice that out here in the extreme values that I mentioned can be hard to predict, they're actually looking much better, both at the high and low ends. And the overall performance within that subset, R squared of 71%, much, much better than what we saw generally across the whole thing. So you can kind of get the idea here that when your model converges well, typically that will come with a better prediction. And in general, if you read the different papers that are out there, the different comparisons, one of the conclusions you'll find is that variable selection methods, such as Bayes C-pi that uses the MCMC process to select predictors, these methods generally perform quite well with oligogenic traits, that is, traits that are explained primarily by just a few genes. So the selection methods are usually able to figure out the right predictors and build a pretty accurate model. Now, of course, there are also going to be examples where methods like GBLUP that are using all of the SNPs are going to perform better. And here I'd just like to show an example of that. This is data coming from Arabidopsis. It's publicly available data that is hosted through the Gregor Mendel Institute, where they have over 1,300 specimens that have been collected throughout the world, mostly in the Northern Hemisphere, majority of them in Europe. But they are inbred lines that have been genotyped on a custom genotyping chip with over 200,000 SNPs available. It's very rich, very interesting data. And as an example of a complex trait, I selected longitude. There aren't a lot of phenotypes available there for the complete data set, but they have the geographic coordinates where each specimen was collected. And I thought, wow, that would be interesting to see if we can identify some kind of genetic model to predict where each sample comes from. And you can see here it's not a normal distribution. Longitude near 0, these are European, North American over here, some outliers that are getting into Asia. Let's see how this works out. In terms of the SNP effects, when we run this through a 5-fold cross-validation, you can see that it's quite different than what we saw with that previous simulated trait. We don't have 6 or 5 clear spikes in the data. We have what looks like a lot of background noise for the most part. But despite what appears to be a lot of background noise, the predictions are actually quite accurate in this case. You can see here at the top, this is within sample predictions, likely subject to overfitting, but it's able to quite accurately build a model that places most of the samples close to their true longitude. But even with the 5-fold cross-validation procedure with out-of-sample prediction, the model that's built explains 83% of the variation in longitude quite clearly. Again, we see some of the extreme values are not predicting well, and of course, there's not much data to base the prediction on when this is held out to be in the test set. The training set's just not going to have a good example. That particular specimen, I believe, was collected in New Zealand, and so also the question becomes, in terms of worldwide distribution, should it be at the high end, should it be at the low end? All we know is that it's the other side of the world from Europe. All right. Now, the message here is that GBLUP and other methods that use most or all of the SNPs are generally expected to perform well with complex, more polygenic traits. And again, that's just a kind of an assumption that I've picked up from my own testing and from reading a few papers on it. So with that, we have a good flavor of how this all works. What I'd like to do now is move out in the software and just walk you through the mechanics of the process a little bit. We'll go through one demonstration with GBLUP and take a look at the inputs and the outputs and see how this all comes together. So we'll go ahead and launch SVS. I expect that many of you have seen it before. If not, just for orientation, I'll point out that when you launch the software, you are prompted either to create a new project, download example data to work with, look at tutorials if you're just getting started, or you can open an existing project. And that's what I'll do today, is open a project where I already have some data. Now, within SVS, we support spreadsheet-style views of all different data types. We can read data literally from 50, 60, 70 formats. I can't remember what the total number is right now. But once the data is in, we support spreadsheet views like this, where we can see 472 rows. That represents our 472 cattle, and over 50,000 columns of data. This representing all of the SNPs available in the genotyping. Also, you see this green map button, which, when I select that, opens this collapsible marker map area of the spreadsheet where I can see some basic mapping data, so I know what chromosome and base pair position each SNP maps to. Now, if I'm interested in doing a genomic prediction experiment, I'll typically want to start by selecting a set of good quality SNPs to base the predictions on. And in this case, what I'll do is, from the genotype menu, choose this option to filter genotypes by marker. And what I'd like is to identify a set of SNPs with very high call rates so that we don't need to worry about missing data. And I'll set it at 99% here for call rate. And we'll look at a minor allele frequency of at least 5%, something that's going to have statistical power to discern between groups in the data. So we'll let this run. It goes very quickly. When it finishes now, we see the listing of call rates, SNP by SNP, allele frequencies, SNP by SNP. Of course, we can visualize those if we want to see what it looks like. It turns out that our allele frequency distribution is really quite uniform in this case, aside from several apparently monomorphic or just low polymorphism SNPs. Now, in addition, we see that the main data spreadsheet has been reduced to 43,000 active columns. Anything that's gray indicates a failure on the filter. and I can make a data subset. So we mentioned SVS has useful data management characteristics. This is one of them. By clicking a button, I can just get a new subset of those 43,000 active columns, and I'll rename it to call it filtered SNP data. Let's close this and look at our phenotype. This is the same phenotype used in that first case study today. And if we want to confirm the distribution on that, we can make a histogram and see that it appears to be approximately normal, which is a very good thing. And what we'd like to do today is select about 20% of these samples to use as a test set, and the other 80% to use as a training set. The way I'll go about doing that is from the Select menu, there's an option to select a subset of the data, and I can specify the fraction that I want. I'll select 80% at random. And you'll notice now 377 rows remain active. Others have been inactivated. And what we'll do now is merge this with the genotypes. Data merges in SVS are quick and easy. I can select the shortcut button here for merging, and it highlights the sheet I'm starting from. I can specify that I want to join it with the filtered SNPs. Click OK. And now we have all the data in one place. You see phenotype 5. If we scroll down, we find about 20% of the data does not have that phenotype because of the filter we applied. And if this is what I want to use as the dependent variable or the outcome variable for any function, I just need to click on the name, it changes color, and that's the visual cue that we're set up and ready to go. Now back here in the genotype menu, you'll notice that down below all of the options for GWAS testing and haplotype testing, there are specific options for Bayesian genomic prediction. That's where you would go to get Bayes C and Bayes C-pi, or the genomic BLUP, GBLUP option. Let's start with that one. In this window, we have just a few selections to make. We need to know, in terms of missing data, how to handle it. I'll select the second option here. It asks about the genomic relationship matrix. If you have one pre-computed, you can specify it here. We did not compute one in advance, so I'll just let it create one on the fly. And then finally, this option at the bottom right is with the missing phenotypes, of which we have about 20% of our data. We want to predict their phenotype and their breeding values, or ignore them and only get breeding values for those with phenotypes. And of course, we want to predict it here today. So we'll let this run. And it's about a 3-step process. In this first step, it's scanning through to determine some background variance parameters. In this second step now, it's creating the relationship matrix. It's quite fast and efficient. And then finally now, it's determining the substitution effects and the sample predictions. So that all finished up quite quickly. We see here 2 major outputs in addition to the relationship matrix. I'll set aside the relationship matrix for now. Also close the original data. And let's focus to begin with on the allele estimates, the estimates by marker. With this output, we see the allele substitution effect. I can sort this and find out which have the strongest negative effect, the strongest positive effect on the trait. Of course, the absolute value makes it a little easier to do that. I can see the strong effects in either direction. And finally, we have this normalized absolute value of the effect, and this is the one that's usually most appropriate visually. So if we want to see what it looks like in genomic space with 2 clicks, oops, that's opening on my other screen today. Let's pull it back. We can see now this pattern that we've already seen several times of apparently strong effects on chromosomes 4, 6, 14, 20, and 29. If you were seeing this for the first time and wanted to explore it a little bit, perhaps starting with this chromosome 6 signal, the genome browse functionality allows you to zoom in and take a closer look at individual SNPs. Clicking on any SNP gives you the information from the spreadsheet about it. But we also have the ability to incorporate other annotation sources. For example, you can see that this SNP is close to a PCDH7 gene. There are external links you can pursue to learn more about that gene and its function. So it provides an interactive option for exploring your data in a little bit more detail. So let's set this aside for now and take a look at the sample-wise output. So here we have an output where sample by sample we see the original phenotype, the predicted phenotype, and the random effect component. That random effect component is the breeding value. It's just another name for it. Now, as we scroll down through here, we see down at the bottom, we have all of our samples where the phenotype was unknown. And SVS assumes, in this case, that anything where the phenotype is known should be used in the training of the model. Anything that is unknown should be predicted. And so we can look at this in different ways. One thing that I'd like to point out while we're here, just mathematically so that everyone's clear on how this works. If you look closely at the predicted phenotypes and the random effect component, you'll see that they're linearly correlated. So if I make a plot of that predicted phenotype versus the random component, or essentially the breeding value, you can see that they're really on a straight line. What happens with this is that that breeding value is based on the SNPs telling the software how much to adjust that sample from some baseline value. In this case, 0. You'll see that if you adjust 0 to the mean value of the actual phenotype, then you'll end up with identical variables here. So it's really fairly simple math to pull these together. Now, if we focus just on those samples that were predicted, let's scroll down and find where those were. I'll turn off everything else and create a subset just of the ones that needed to be predicted. I'll now merge this with the original phenotypes so we can get an idea of how well this performed. In this case now, I can make a scatter plot where I select that original phenotype 5 for the x-axis, select the predicted phenotype for the y-axis, and plot this. Now, initially, that may look a little bit random, but if we add in the actual diagonal regression line, you can see that there's definitely some trend here. If we go back and run some other statistics on it, we'll be able to find out just how accurate the predictions were. And we can definitely see that some of the highest original values are definitely among the highest prediction values. And given that we have a relatively small sample size here, I think that this performance is probably quite good. So hopefully what you've seen in this very brief demonstration is that SVS is able to provide a unified environment for working with genomic data in different ways, doing simple quality statistics and doing very advanced visualizations, along with some advanced genomic prediction work as well. So let's come back to our slides here for a moment. And just to wrap up and repeat once again, genomic prediction techniques provide us with many things. Among them, we can predict phenotypes, we can predict breeding values. This helps us to identify which animals or which plants are most likely to pass on desirable traits. And genomic prediction also can tell us about the influential loci for a particular trait, which loci can be used to develop targeted assays or diagnostic tests, and so on. And SVS gives us a unified, integrated solution for managing all of that data, for running genomic predictions, doing association testing if needed, and doing visualization of all of those variables and all of that data within a single space. Now, if you'd like to learn more, we have a lot of additional resources on the topic. We've recently released an ebook by our CEO, Dr. Andreas Scheer, called Genomic Prediction in Agriculture. You can download that from our website at goldenhelix.com. Greta Peterson, our senior biostatistician and director of product quality, gave a webcast on this topic several months ago. That is recorded and available on the website. It has a little bit more technical detail on some of the algorithms involved and is another excellent resource. Also, anyone who's attending the PAG conference next month in San Diego, we will have an exhibit there, and we would love to visit with you. So please stop by. So if you have any more questions or want more information, you can visit our website, goldenhelix.com, download those other materials or request an evaluation of the software, or send us an email. We'd love to hear from you. So with that, I think we will get ready to answer some questions. And I'll start with this one. Does the program allow fitting fixed effects in GBLUP? The answer to that is absolutely yes. I didn't really focus on it in the interactive demo, but there was an option there to add additional covariates into the model. And any numeric or binary variable or categorical variable, I believe, can be accounted for in that manner. A similar question, is it possible to use several phenotypes in the analysis and get prediction based on a combination of those, potentially with specific weighting? That's going to be a bit more of a challenge, but if you can incorporate those additional phenotypes as covariates, then that would be probably the best approach to take. Does the Bayesian method as implemented in SVS account for relatedness? Yes, and we didn't walk through the process for the Bayesian method, but similar to GBLUP, it incorporates a genomic relationship matrix. I believe there was another question here that said, or it asked if there are options available for calculating the relationship matrix, and yes, I think we have 3 or 4 or 5 different methods at this point. We can do the identity by state matrices, identity by descent. We can do a G matrix, or there's a matrix. There's several different options that are in there, and be happy to talk more about those offline. So another question, should the phenotype distribution be normal? And if not normal, how to do? That is an excellent question, and typically I would say that the prediction accuracy is going to be best with normal or near normal distributions. If you have a binary trait, the methods as implemented in SVS will work on those, but the accuracy, what I'm saying is it's not going to assign a 0 or 1. It's going to treat it as though it was numeric. Moving down, let's see, can exome data be used instead of SNP chip data for human near Mendelian disorders? That's an interesting question. It's a case that I haven't really tested. The question becomes, with exome data, typically a matter of the frequency of the SNPs and what kind of statistical power you get from them. Quite often, the SNPs on the exome chips tend to be very rare, very low frequency, and as a result may not be ideal for model building, but certainly there's nothing to stop you from doing it if you want to give it a try. So another question, was there any bias in estimation of SNP effects in GBLUP and BASE C-pi as often seen in single SNP estimates? That is an excellent question. And certainly, if you look back at some of those plots that I showed in terms of the actual versus predicted phenotypes, you will notice if I were to fit a regression line that there would be a very different slope. And you might say, well, maybe the predictions are biased a little bit at one end or the other. And if you were to dig into the details of it, you may find that there are individual SNPs that may have unusual influence, although generally I would say with GBLUP, given that it fits so many SNPs into the model, it's hard for any one SNP to really influence things. But we did see the one example with BASE C-pi, where the R implementation and the SVS implementation selected different SNPs. It was very influential for the few samples where those SNPs had different phenotypes, or sorry, different genotypes available. So it's something that you should watch out for. Question, does SVS use all markers to predict breeding value for oligogenic trait? That depends on which method you use. GBLUP is going to use all markers, or at least all of the markers that you as the user provide to it. So you saw in the example that I ran, I selected markers based on allele frequency and call rate. I can see situations where you may want to restrict further and only give it as a starting point SNPs that have some prior association to the phenotype perhaps, in which case it would only use what you give it. Now BASE C-pi, of course, whether you give it a lot of SNPs or a few SNPs, it's usually going to try and identify just a few of the most influential SNPs to build the model. Okay, so with that, I think we've covered most of the questions. There are a few more, but they all appear to be fairly similar to questions we've already answered. So I think we'll go ahead, Cheryl, and wrap this up.
Thank you, Bryce. Any remaining questions, if we missed any, skip them. We will get back to you for certain, and feel free to contact us in the meantime. Thanks again for joining us, and have a wonderful holiday.